So we're going to go ahead and get started. I'd like to welcome everyone to the Vasculitis Foundation's webinar today. I'm Kathy Olevsky, and I'm the host for the Vasculitis Foundation Educational Webinar Series. I'm also a patient living with MPA vasculitis, and these webinars are part of the Vasculitis Foundation's commitment to patient education. We'd like to thank our sponsors, AstraZeneca, Amgen, and Novartis, for supporting these webinars. Today's webinar is CAAR-T cell therapy with Dr. Megan Free. And after we finish this webinar today, there'll be the recorded version that I was telling you about earlier on the YouTube channel, and everyone will get an email as soon as it's ready. And now that all of that housekeeping is done, I'd like to introduce our guest for today. Dr. Megan Free is an associate professor of medicine at the University of North Carolina at Chapel Hill, where her research studies the role of CD4 plus T cells in autoimmune kidney diseases. One particular project is focused on detecting and understanding autoreactive T cells in ANCA vasculitis. This is balanced with studies to understand regulatory T cells in vasculitis and how to restore their anti-inflammatory functions. Her studies provide insight into potential causes and predictors of relapse from an immunological perspective. Welcome, Dr. Free. We're so happy to have you here with us today.
Thank you, Kathy. It's great to see you again. Some of you may have seen me about a year ago. I spoke at the Vasculitis Foundation in person in Chapel Hill, and then we kind of had a follow-up webinar about emerging therapies in the vasculitis field. So this is kind of an even more closer look into some emerging therapies in this field, and I'm always happy to come back in and speak with all of you about where science lives currently and how that may impact the medical field, and particularly vasculitis. So I'm definitely excited to be here today.
Well, we are so excited to have you here. I did need to ask you before you get started, do you have any disclosures to share with us today?
I do not have any disclosures.
Okay, and if you'd like to share your screen, we'd love to learn more about CAAR-T cell therapy.
Sure thing. Can you see it?
We can.
All right, perfect. I am going to pull up the laser pointer just in case. So I'm going to use some of these slides more or less to kind of help guide this discussion. I really look forward to the questions that you all may have at the back end of this. But I do want to kind of lay the foundation and the landscape of the immune system so that we can understand that before jumping all the way into CAR-T cells. So within your blood, there are numerous different types of cells that are circulating, both red blood cells, which are also called erythrocytes. We have platelets, which help clotting. And then the rest of these are all white blood cells. So we have, neutrophils, which are the targets in ANCA vasculitis because they're the ones that house MPO, myeloperoxidase, and proteinase 3, PR3. But the ones that we're going to focus on today for the purposes of CAR-T cell therapy are lymphocytes. And lymphocytes is kind of a broad term because you can break down lymphocytes into numerous subtypes. So you may have heard of B cells before. These are lymphocytes that produce antibodies in our blood. And these are really important to protect us from infections, viral infections, bacterial infections. But you know that in autoimmune disease and in ANCA vasculitis, there's some B cells that have gone awry. So B cells have started making antibodies against your own self proteins, and that is causative in disease. And so a lot of the drugs that are utilized in vasculitis, but also in many other autoimmune diseases, do target the elimination of B cells. But many of these drugs are kind of very broad. They target all B cells, so good B cells along with bad B cells. So one of the things that we look for in science and research is how can we be more selective? How can we just target the bad B cells or the ones that are producing antibodies that are causing disease? Now, balanced with B cells are numerous different types of T cells. We have CD8 T cells, regulatory T cells, and CD4 T cells that all have different functions and various targets, but collectively, all these cells work together very nicely to provide immunity against various pathogens we might encounter. But when we get into CAAR-T cells, we're looking at these CD4 and CD8 T cells are what we're going to talk about today. So they're a type of lymphocyte that is circulating in your blood. I'm going to talk about both CAR CAR T cells and also CAAR T cells. Both of these have been engineered for different purposes, and we'll kind of walk through what they are and what are the differences between these two different types of CAR T cells. So the first type I'm going to talk about are kind of the traditional CAR T cells with just one A. And what does CAR actually stand for? It stands for chimeric antigen receptor. And what that means is that these cells have been engineered genetically to recognize a very specific protein. So normally what happens to T cells in our blood is that they're educated in your thymus and they kind of have a random assortment of what their receptors are going to be. And this is how we're protected against any and all bugs that we could ever encounter, because genetically it is random as to what a T cell's receptor is going to be until they're educated. What the engineering of a CAR T cell does is it basically forces that T cell to recognize one certain protein. And the reason why this is important and why it was developed in the cancer field is numerous cancers have very specific proteins on their surface that are different from healthy cells. And so it provided this opportunity for T cells to be engineered against just those tumor antigens and eliminate those while keeping the rest of the cells safe and healthy. So typically what happens in this process is that the patient's own T cells get removed from the blood. These T cells then go to the lab in which a gene for the chimeric antigen receptor is inserted into the T cell. Once we know that that gene has been accepted by the T cells will proliferate. They'll grow into millions and millions and millions of T cells in a dish in an incubator in the lab. And then once they go through a series of safety checks and kind of profiling of those T cells, they are then infused back into the patient. And it's within the patient that the hope is that these CAR T cells are going to seek out just those cells that have that antigen that they've been programmed for on their surface. So in the case of cancer, these CAR T cells are searching for the cancer cell that has that particular antigen on their surface and will cause those cells to die. So this is how in cancer you get a very selective reduction in cancer cells instead of kind of broad spectrum chemotherapies. Now, what is not shown in this process, it's nice when we can kind of distill this down into one little slide, is the amount of time that it takes to get from taking a T cell out of a person to, genetically manipulating them in the lab and growing them up in the lab and then giving them back. It's an extensive, laborious process. And so when this is done as a therapy for a patient, it's done on a per person basis. This is not something that we can get off the shelf and give to a patient. It's a very manipulated process that is done on a person by person basis. So in the cancer field, it has been most frequently used for B-cell lymphomas early on. The number of cancers it's being used for is expanding. Each week and month and year, there are more and more cancers that are utilizing CAR T cell therapy. And currently, to date, I just checked the most recent literature, there are 7 CAR T cell products that are approved by the FDA for cancer. So those 7 therapies that are approved in cancer, now this is, FDA approval versus there are plenty of other CAR T cells that are in clinical trials that are different from this. But those that have been approved, their kind of scientific name is here. Then what you would see them probably more advertised would be this name. And then what the target for the CAR T cell is here. And you can see that the early generation of CAR T cells were either generated against CD19 or BCMA. Both of these are proteins that are found on B cells. So these are CAR T cells that are going to seek out and selectively kill off B cells. And that's why the vast majority of these were initially marketed for B cell lymphoma. Now, I will say the evolution of CAR T cells has really come a long way. We're up to 5th generation CARs at this point. Originally, what the first generation was, I don't want to say simplistic, but it was proof of principle. Can we, engineer T cells to seek out a certain target and be successful? And the answer was yes. But what you can see is that typically, what is on the outer side of the CAR T cell has stayed the same. It's what is within the T cell on this half that has changed over the years. And all of these, what they have done is added more specificity to how the CAR T cell works. So some of these are what we call signaling cascades. So they help activate the T cell whenever they get to the right place, when they get to the right cell to kill. And then some of these, like the one that's called an IL-12 inducer or an IL-R beta chain, these help the T cell function in the correct context. So it helps them release proteins and cytokines to help kill off their target more efficiently and more effectively as compared to the 1st and 2nd generation CARs. Now, the other interesting T cell that has been engineered are chimeric or CAAR T cells. So some people call these chimeric autoantibody receptors. Some people call them chimeric autoantigen receptors. It really depends on what is engineered to be on the surface of the T cell. But this came about from more of the autoimmune perspective. People within the autoimmune field looked at the CAAR-T cells and asked the question, can we utilize this technology to our advantage, but kind of flip it on its head and seek out autoreactive T cells instead, or autoreactive B cells, instead of seeking out cancer cells? So this is kind of the nice comparison, and they initially did this in a model of pemphigus vulgaris, which is an autoimmune skin blistering disorder. So, this is what we just talked about, the chimeric antigen receptor, in which you can generate a CAR T cell against CD19 that will go and selectively kill off any CD19 expressing B cell. In this particular study, what they did instead is they had the CAR T cell express the autoantigen. So in pemphigus, the autoantigen is known to be desmoglein 3. And what these T cells then go do is they seek out any B cell that might have an antibody that is recognizing this autoantigen and will selectively kill off just that subset of B cells, leaving the rest of the B cell population alone and functioning normally. So what you can imagine here is, in the case of, say, ANCA vasculitis, if you could generate a CAR T cell that is expressing myeloperoxidase or proteinase 3, would these CAAR T cells then selectively seek out and find anti-MPO and anti-PR3 B cells and selectively kill those off while preserving the rest of the B cell population? So what I want to talk about a little bit, though, is that there are definitely pros and cons with CAR T cell therapy, because probably at this point you're thinking this all sounds wonderful. When can we get some? And, selectively just kill off those bad B cells. But these are not without some risk. And definitely we have a lot of data from the oncology field that has helped kind of steer the CAR T cell field. And also, as you saw earlier, there's been multiple generations of CAR T cells that have come along. So we're definitely improving the technology as the years go by. So in terms of pros for CAR T cell therapy, I think one thing that you can definitely appreciate is that these CAR T cells are customizable, meaning that we can generate CAR T cells to recognize and go after, cells that express any sort of antigen. So there's definitely a potential for more selectivity. And about a year ago, there was a New England Journal of Medicine paper in which they did use CD19 CAR T cells in patients with lupus, in which their lupus was severely refractory. And it was successful at inducing longer remission in those particular patients. And I have, a little asterisk here that this therapy in that cohort of patients was longer lived than rituximab. And one of the reasons why there's speculation this might be is that the antibodies of rituximab do eventually get cleared from your system versus CAR T cells, once they're in circulation, may last longer. And some of them may turn into memory cells that could keep some of those cells at bay. Now, in terms of cons, one big one is the side effects. In terms of the side effects for patients with cancer that get these CAR T cells, cytokine storm occurs pretty frequently. And what cytokine storm is when you have that many cells that are being targeted and killed, there is a release of proteins associated with that can kind of stir up an inflammatory response. And that's that cytokine storm that we're talking about. One of the things that's a little more concerning, but it's really at its infancy, so it's kind of one of those watch and see situations, is that same New England Journal of Medicine paper, or that same edition of the New England Journal of Medicine, also had a paper that, was a cautionary tale that some of these patients who were originally treated for B-cell lymphomas, while that particular cancer went into remission, they, several years later, were reporting secondary cancers. Now, this is not across the board. This is a minority of patients. But what is a little bit cause for concern is that it was not their original cancer coming back. It was a T-cell lymphoma. And some of those T-cell lymphomas that they were able to genetically sequence did have the CAR gene within them. So there is, kind of some eyebrow raising in terms of, what is the oncogenic potential of a CAR T cell? And, as researchers, do we need to rethink the engineering of those cells? So as I mentioned, most of the CAR T cell work has actually been in the oncology field, but we are kind of on this precipice of utilizing CAR T cells in autoimmune disease. And I wanted to give you kind of a taste of where are we in that particular situation. So as I mentioned, this New England Journal of Medicine paper last year, it was a case series in which they used the CD19 CAR T cells. So these are T cells that are going to seek out and kill off any B cell that is expressing CD19. So it's a very similar mechanism to rituximab. The patients who were included in this case series either had severe refractory lupus, inflammatory myositis, or systemic sclerosis. It was only a total of 15 patients. The majority of them did have lupus, but they did have good success in inducing remission in those patients. And you can imagine for patients who have very severe disease that have failed multiple immunosuppressive regimens, this was the reason why they were included in this trial. Now, I will say from a very specific ANCA vasculitis perspective, there's a paper that came out last year. It was actually presented at the vasculitis meeting in Barcelona last year, that they utilized CD19 CAR-T cells in the ANCA-GN mouse model. Now, this was a model of prevention. They gave the T cells prior to induction of disease, but it did prevent onset of disease in those mice. And then there was a study that just came out this year in 2025, in which this particular group treated just one ANCA vasculitis patient with CD19 CAR T cells, and they did have good sustainable remission. So I say all this to put it out there that this is a thought process that is occurring, and it is being used in very select instances. And really to highlight that, and anyone can do this, you can go on clinicaltrials.gov and search to find, what clinical trials are out there, who's recruiting, who's not, what country is the clinical trial in, what are the criteria. But when I looked at it the other day, there are currently 30 different CAR T cell clinical trials just in the United States for autoimmune disease. Now, I will say a good portion, greater than 50% of these, are targeting lupus patients. And part of that is just because the outlook for lupus is so dismal in terms of how many drugs are not approved for that patient population. They desperately need a new therapy to be approved for that particular disease. But I wanted to highlight that there is a study that is recruiting ANCA patients for allogeneic CAR T cells. Now, I want to highlight what allogeneic means. So that image I showed you earlier, in which the patient is giving their own T cells and then waiting for them to expand in the lab and then giving them back, that's called autologous. So auto meaning self, you're giving the cells back to yourself. Allo means that the cells came from someone else. So what we are seeing in research is that there are ways to generate allogeneic cells that are kind of stripped down of anything that would be seen as foreign by the other person. And so it's a way to have a more off-the-shelf therapy. So it would be a shorter time to be able to get to those T cells. There's another study that's including ANCA vasculitis that, again, these are allogeneic cells, but they're utilizing CD20 gamma delta CAR T cells. Now, without going too deep into the weeds of immunology, the vast majority of T cells that are circulating in all of us are what are called alpha beta T cells. Gamma delta T cells are a very small percentage of our T cells, 1, 2, maybe percent. They're a little bit different, but it's interesting that they're utilizing them for this particular study. And then there's a third study, also including ANCA vasculitis, that is a much more traditional autologous CD19 CAR T cell therapy. So I want to kind of end with that, just to point out and show that this is definitely a pivoting point within the autoimmune field, that there are clinical trials ongoing to look at this, to utilize this, and see what the risk-benefit ratio is here, and which particular patients would this be most beneficial for. So I think it's an exciting time, both for researchers and for patients, to really start getting towards more personalized medicine and more selective with our treatments and therapies.
Thank you so much for that presentation. That was extremely interesting. A lot of science things in there to learn. And you can stop sharing your screen so we can see your face again. There you go. We have had a few questions come up, and I would like to remind everybody to please put your questions in the Q&A box if you think of them as we're going along. I was going to go ahead and ask the ones that have come up so far. And one is, it looks like right now it's a long process with this technology. Do you see it evolving in time where the amount of time it takes to get the cells out of the patient and grow, will that improve time-wise, or is that always just going to be a very long process?
So I think with autologous CAR T cells, so coming from the patient themselves, that's always going to be a longer process. I think, there have been improvements in the protocols over the years that it's gotten shorter, but it's always going to be a longer process. Just by virtue of it takes a while for your cells to divide enough in the lab to have kind of a critical number to give back. What will be quicker and closer to off the shelf would be the allogeneic route. So, having cells from someone else, a healthy donor, that they engineer and kind of keep in a bank that they can, give to patients more on demand. And we're seeing that more with what are called like CAAR-NKs. They're NK cells, so another type of immune cell that is more allogeneic friendly. So there's, from yourself, it's always going to take a while. From another person, it's going to be quicker.
Okay, well, that makes sense. And could this, I guess this is all still, these questions are very up in the air, but we want your opinion. So I'll ask, could this potentially, you see this helping vasculitis patients? I mean, I know you said it can last longer than like rituximab. So is this something, are we far out from possibly helping vasculitis patients?
I mean, I think we are much closer than we were 2, 3, 4 years ago. Probably 4 or 5 years ago, the concept of using a CAR T cell in any autoimmune disease was kind of a pipe dream. And now you can see, just in the US alone, there are 30 different clinical trials using them in autoimmune disease patients. Um, from the CD19 CAR T cell therapy, I personally feel it's a little bit of a wash between rituximab versus CAR T cell. I think rituximab's probably still, we have more long-term safety data on. I
Mm.
think where the potential for a lot of help comes from is the specificity that could come from CAR T cells. So if you could engineer a CAR T cell to only target those anti-MPO or anti-PR3 expressing B cells, um, and just kill those off while preserving the rest of your immune system, I think that's where kind of that holy grail lives. Because a lot of the immunosuppressive drugs now, you're more susceptible to infection because it's dampened, kind of unilaterally all your B cells and all your T cells. So getting to a point where we're only targeting the bad cells while leaving the rest of your immune system intact is that sweet spot that we all hope to get to.
Yeah, wouldn't that be awesome for all of us patients
Yeah.
to not kill off everything that we have? Speaking of that, can these T cells be taken from any patient, or is there like a specific criteria?
So without getting too into the weeds, I don't know all the specific criteria from like a clinical trial, but I mean, I do think that they have criteria in which you have to have a certain T cell count to start with so that you have something to pull from in order to engineer. But another thing that's not talked about a lot, there are clinical trials that do this and some that don't, is some trials do what's called preconditioning. So they're kind of drugs that do wipe out part of your immune system so that when they give the T cells back, they have a better chance. Um, so, there are additional kind of stipulations to, is the patient the right patient to undergo this particular therapy?
Okay, and I know a lot of this is just information that you're just now learning and presenting, but we have questions.
Yeah, definitely.
One of the questions that did come up is there was some conversation, and of course we latched onto that there were some vasculitis patients in some of these. But it said ANCA. Were there any specifics on what type of vasculitis, or was it just an ANCA positive patient?
So I mean, from the clinical trials that I was looking at just the other day, they were inclusive of both MPA and GPA. I've not seen anything specifically looking at patients who have eGPA.
Okay. And let's see. Would this perhaps be an alternative for those experiencing breakthrough flares whilst on rituximab or another current therapy, i.e., someone who can't go properly into maybe refractory, somebody who can't go properly into remission?
So I mean, this is all speculation because I'm not with any of these clinical trials and I am not a physician, but most of these clinical trials are very geared towards those refractory patients. That is the patient population that they're after because they are the ones that are likely to benefit the most, and it's where the benefit is the highest to outweigh the risk.
Interesting question here. Is this only for adults? Any knowledge of this will be something to use if JC virus is detected in the system.
So I actually don't know anything about potential pediatric data.
Mm.
I mean, I can definitely look back, but if there's any pediatric data, it would be from the oncology field. I would seriously doubt that they would have any pediatric patients in the early stages of these autoimmune clinical trials. Um, and concerning like any sort of like, if you have high viral loads of something, I would doubt that they would be a good candidate for the CAR T cell, just because, as I mentioned, a lot of these trials do preconditioning, which kind of wipes out part of your immune system. And if you already have high viral titers or viral loads, and then you wipe out your immune system, you're at risk for that virus kind of taking over. So it would have to be someone who had, stable viral load.
Yeah, that makes sense. Any concerns with genetic engineering aspect of CAR T cell production? Because you know that comes up all the time.
So, I mean, I touched on this a little bit in the con list of the CAR T cells. The very few patients, again, it's a minority, but it has happened. Those patients who had a secondary T cell lymphoma in which the CAR T cell gene was detected, that is a little bit of a red flag to me. Is that just that particular person had already had cancer, and so their body is more prone to cancer in general, versus did something happen in the engineering of that T cell that made it more prone to having oncogenic features, meaning, more likely to become a cancer cell. I think those are unanswered questions, but they're definitely concerns that need to be followed up on and understood better.
Okay, that makes sense, and that's a controversial subject. So, um, there's a question. I honestly don't know what it means, so I hope it's related to CAR T cell. Could you comment about George Schett's groundbreaking work with long-lasting remission off therapy for patients with other autoimmune diseases? If it's not CAR T cell, you can just say that, or if you don't know.
I don't know.
Okay. Well, thank you, Ida, for asking that. Well, maybe that's something we'll look into. We don't know
Yeah.
the answer to that. Um,
It
you,
is CAR T cell therapy.
Okay. I can look it up and swing back around.
okay. And then let's see what else I think that's all we have in the actual questions. There was a question about could you name the 7 products that are currently FDA approved? And you did that in your presentation. The other
Yeah.
part of his question was what cancers are they intended to address? But that it seemed like you answered that. I don't know if you have anything else you wanted to add to that.
Yeah, so I mean, those 7 that are all approved, they're against CD19 and BCMA. So their primary indication for most of those is for B-cell lymphoma. I think some of them have been used for like mantle cell lymphomas and kind of other B-cell derivatives. But that's the main target of those CAR-T cells.
Okay, well, I at this point will say thank you so much for everything that you shared with us today. I felt like I'm not a science person and I totally understood that presentation. So maybe that's a backhanded compliment. I hope you will
No,
take it that way.
no, it's always great when everyone, takes something away from it.
Yes. And I also want to just say thank you to the Vasculitis Foundation, of course, for supporting these webinars. And maybe just actually before we get into that, I want to talk just at the end here about how we as patients can actually help with research, since you're talking about research today. And one of the ways that we can do it is by joining the Vasculitis Patient Powered Research Network. And I am actually a patient champion for the VPPRN. I encourage other patients to join the VPPRN and to complete their biannual forms. And speaking of that, I think as of this recording today, there's like 2 days left to complete your biannual form. So if you haven't done it, look in your email. You should have gotten an email. And the deadline is March 15th. So it's 10 minutes, I swear. Just go do that. And I can't tell you everything about VPPRN, but if you'll visit their website at vpprn.org, you can learn more about their patient registry. They have a dashboard that tells you how your information relates to our particular vasculitis community. It's really, really a great process. So we would like to thank the VPPRN for being available to us patients. And also, of course, thank you, Dr. Megan Free. Thank you to the Vasculitis Foundation and our sponsors, Amgen, AstraZeneca, and Novartis.